Oh no, I'm thinking of first by the way, I was thinking maybe we could do like 2 Oh my god.
But we also got a lot of beers.
That's where I'm gonna be.
So first time you circling
Exactly.
Oh yeah.
You're supposed to be recorded.
This is a local guy. We're building all the time.
Nice.
It's different than, yeah. Yeah, I remember getting that email from David where because Yen was corresponding with him about seminar speakers that morning, and so he shot a reply back and said, I'm in Santa Cruz today, you know, but I'd love to get somebody who was visiting on, I'm like, you're in Santa Cruz today? You're supposed to be here. Yeah, no, it should be good. Yeah. Yeah. Until that's
Funny, you still have till tomorrow. You still have till tomorrow at 5.
right there.
Good? Yeah. I did this morning. Also, watch that soccer highlights.
Can I put my intro on your? You'll have to move away so you can't see it.
Okay. First things first. Not just defending.
Just say all here?
Yeah. First is
Have CNs.
Judge Miriam.
I have no idea. Most hours out. Most hours out.
Hey, I'm going to do plenty of pauses to gather myself. So I did, thank you. I don't know if I used any of them actually, but I'm very appreciative of those because I want to. Yeah, I started developing a lab photo art. How are things going for you, Kat? Yeah?
Working on my acknowledgements. Very, very short. Even thank Drew for not joining the last one.
Just to begin.
Yeah. John's parents like my poster better than this.
I didn't even like the better, but they asked me why mine wasn't so good. Wasn't as good for us.
Well.
Which was like a great thing to happen to
You'll have to go sell your body for it.
I mean, I think.
I had 1 year never
Sucks it up.
Oh,
You've gone along with it.
I gave my thesis.
Did you really?
Yeah, and so I mean, it's like, yeah, it's good. I don't think practice. You're right. My whole, I think my whole, there was no, I mean, I was fact that aliens, are black. My laptop was not handled. It was like a 96 megabyte. And I was like, some are
Oh, thank you so much. Yeah, I'm emailing, trying to figure out how to send out the invite now. But thank you so much for all your instructions. They're perfect. So, Yeah, that should be okay.
Okay, welcome everybody. It's a pleasure to introduce Jonathan Lynch for his thesis defense today. Big day for John, big day for me. It's a pleasure to have his parents in the audience today. Thanks for coming. They're all the way in from the East Coast. His girlfriend Christy is here. And then several people are watching via live stream, which is exciting. And thanks to all of you for coming, committee members and colleagues, friends. Thanks for being here. Of course, it's a huge day for John. Many years of work you'll see presented today. I'll crunch down into 1 hour. This is also a big day for me. John was the first graduate student to join my lab way back. And you know, he was an incredible first graduate student. He set a really great tone in the lab. He worked really hard, also has a great sense of fun, and totally set up the lab culture, I think, in our group. So I can blame that on him. Yeah, so he joined the lab in 2009, and you know, came to the lab bright-eyed, enthusiastic, eager to learn. And this is 6 years later, hardened by science. But it's an equilibrium. Depending upon the day or hour, things can change rapidly. This is the picture on the far side is really more representative of what he's like most of the time. But in any case, definitely his style has evolved while he's been in the lab. And so, yeah, so he came to me wanting a rotation project right when we were setting up the lab, and it was great to have this new potential grad student, and I was eager to have him, but we had no mice, and much of the work in our lab, as most of you know, revolves around our gnotobiotic mouse facility, and so I had to come up with a project that was mouse independent for him, and there was this really important question studying a class of novel sensor regulators in anaerobic bacterium of the gut. We knew very little about these molecules, and they were fascinating to me, and I wanted John to study them for his rotation. And what I really wanted was to figure out how to trigger this sensor. You can see the question mark up there. So we could study all these interesting intracellular domains, how these signaling systems were working, but we didn't know how to trigger this, what the ligand was. And so I suggested to John that he actually take a sensor domain that we actually knew the ligand for, and to actually use that as a disguise, and to paste that onto this other sensor that we wanted to study. And that way we could trigger it and then study all the interesting things with the inside of the signaling molecule, not knowing any better that its outside was disguised with this other domain. It was a long shot. I think it was like, you know, I estimated the odds at 1 to 100 at the start of this project, but I thought at least by the end of it I'll know if he can do molecular biology, if he can make this molecule splice pieces of DNA together. And so I pitched this project to him. I said, do you think you can do this? And he gave me the thumbs up. He said, yeah, I can do that. And I mistook this for naivete at the beginning, but this was really his attitude throughout grad school, that he just has an incredibly optimistic attitude, and he works incredibly hard to accomplish goals, and set out to do that with this project. And so the end of his rotation came about. He had actually made the molecule, so he had successfully done the molecular biology part, but he couldn't get it to work. And so I went to him and I said, okay, you've done a great job in here, and at this point he wanted to join the lab. I said, you're in, this will be fantastic. But we have to find a new project for you. And he said, no, I'm going to get this thing to work. And he had some ideas about how to take away regulatory roadblocks to actually get the signaling molecule. And I said, okay, those are reasonable ideas. You know, I didn't want to kill his enthusiasm. And so I said, you have till the end of the summer to get this thing to work. And so this is an email from July 14th, 2009. He says, I hope you and the family are having a good time in Massachusetts. He points out that I'm on vacation while he's in the lab working. And he says, I analyzed the data from the qPCR I did last week. I think I got some interesting results, but and this is so typical of science in general, but definitely of new grad students, that there's always a caveat with good results. But so over the following 24 hours, he worked really hard to figure out what the problems were, and this is the following email thread. I said, am I reading this correctly? He got this incredible fold change in his experimental conditions, and he says, I believe this is correct, and I said, great. So he had effectively set up the foundation for this really fantastic project and was off and running. And so it's not really surprising that he got a disguise, a molecular disguise, to work. You know, if you know anything about John, you know that he's really good at disguises. These are pictures from all of his Halloween costumes. You know, sometimes creepy, sometimes menacing, but they're always great. One year, so this upper right one is really great. So this is Catherine Ng, another grad student in the lab, who will be defending actually tomorrow at the same time. I'll be here all week, so. So they dressed up as each other, and not only dressed up as each other, but impersonated each other, which was really fantastic. This isn't even actually a Halloween picture. That's just at our retreat, John in his pajamas. So pretty funny. But something else about John that is readily apparent in minutes is that he loves sports. He's if you walk into the lab, he's always dressed, ready to go play something here. So this may look like another costume, but this I think is actually a soccer outfit. But maybe not. I don't know. But we would play basketball on Thursdays outside. And in fact, he scheduled the defense today to conflict with this basketball game. Some of his committee members still made it out there to play. I did not, and I thank him for that because it saved me from getting embarrassed yet another week. But he, you know, frequently would dunk on me out there. Very embarrassing. And this was, they actually post rules out there, and you can see the last rule here. This is strictly forbidden, and yet he still did it. But also, if you look down, there's an emergency number there. It's interesting that it's a 310 number. But there is an emergency number, which is good because the other thing that John was known for in grad school was constantly getting hurt. He was just always injured and always on crutches. And actually, if you look at Kat's impersonation of him, she has a crutch because he was so often you know, hobbling around. And in fact, playing basketball, he actually, just this past year, dislocated his shoulder out there while we were playing and had to talk me through resetting it, which was something I'll never forget. Very but I was happy to help after all that he's done in the lab. But there's really a long list of injuries. He tore his ACL. He slashed his hand with a culture tube. He had IGS at one point. Fecal clearing. And that's just a partial list. It goes on and on. But in addition to the pain and the injuries, there, of course, were a lot of good times, and usually John was at the center of it. In many of the photos, he's blurry, actually, because he's doing something crazy, and everybody else is laughing watching him. This is a skit he was doing at our lab retreat. This is at our lab's beer tasting that we have every once in a while, and I think he's impersonating Kat here. You can see people are enjoying it. Here he is cracking Jessica up. Something with a mini Santa that he got as a gift. I think he's trying to get on the naughty list there. But it's not just people in the lab. He's actually incredible around kids. So at lab functions, he's usually playing with, entertaining kids, sometimes terrorizing them. But it's always in good fun. And this is really a gift that he has, and he's actually used it for really constructive purposes. He's helps out with teaching kids science a lot. So this is him and Kat volunteering at an elementary school science fair, and he's tutored throughout his entire graduate school career to help younger kids learn science, which has been pretty great. And so definitely his teaching is one of his superpowers that I see. He has, again, a long list of superpowers, but these are some other ones that I thought were really noteworthy. He can generate momentum on really difficult problems. So if you remember when Superman tried to turn back time on Earth, he had to fly around it several times to get it spinning the other direction. And this is like what John does in the lab. He picks a really difficult problem, works incredibly hard, and you gradually start to see things move, and it's pretty exciting. He's fierce and bold. He'll stare really difficult problems in the face and take them head on. And because he was the first member of lab, I really credit him as being one of the keys for recruiting this incredible group of graduate students and other scientists that we've assembled in the lab. And so he's going to be greatly missed when he leaves the lab. Luckily, we have him for a few more months, so we don't have to deal with that yet. He's interviewing for postdocs now. He already has a great offer at Ned Ruby's lab in Madison studying squid vibrio symbiosis, which is a place I'd like to go do a postdoc, so I'm trying to convince him to switch with me. But anyway, I won't delay any further, and I'll hand it over to John.
So you think by now I wouldn't pose for pictures anymore, but unfortunately that's something else I forgot in grad school. So I'd like to begin by just saying thank you all so much for being here. It means a lot to me. I am always surprised and appreciative of the support I get from people like this. So thank you. So I guess my poster was a bit vague, and I'll try to make it a little bit bigger with my other title poster. But today I'm going to give you a little bit of what I've been doing for the past couple of years. I'm going to try to start kind of at the beginning and finish at the end, but it's a bit of a non-linear progress, so just try to stick with me, okay? So I'd like to begin with just kind of laying out what I'm going to do. I'm going to, like I mentioned before, I'll give you a brief introduction to sort of the general problems and ideas that I've been working with. Then I'll talk a little bit about some of the work we actually discovered, and I'll try to finish up by sort of a project that I guess this is like when Superman got shot with kryptonite, where he was like, oh, this really hurts, and, oh, and then he won, and then he kind of died, and then he didn't die. So I'm not sure what part of that I'm in, but it's been a stumbling block, but something really exciting, really looking forward to working more on. So just to begin with a brief introduction to the microbiota. If you've heard anything from our lab the past couple of years or have heard of the microbiota, you know, it's kind of this, it's been kind of this eye-opening experience for humankind to realize that we're not really people. I mean, we'd like to imagine we were people. It's really cute to think that we're this apex organism. It's really complicated. But really, when you get down to it, we're kind of a walking, talking, breathing, war-fighting group of bacteria. So the fun number we like to present is that if you look just at the bacteria in your intestines, which make up the community, I'll talk a lot about the intestinal microbiota, there's probably about 10 times as many bacterial cells in that one environment on your body than you have human cells in your entire body. So it really looks more like this, with this being our microbial community in our intestines, this being you. And then within that community, there's hundreds of species with tons of different genes. So if you look at the consortium of genes in your intestines, you're actually probably closer to this. This would be human genome, this would be bacterial genes inside your intestines. So really we're becoming better able to appreciate the complexity and diversity and sort of enormity of the bacteria that live in and on us. Now, we've known about this bacterial community for a long time, but really sort of the effects that it has on human physiology we've just been able to appreciate more recently. So this is a non-comprehensive list, or I guess organizational chart of different areas that the gut microbiota alone influences. And you can see here it spans from lungs to the brain, to the liver, to bones even. And now you might have even seen sexual articles where the intestinal microbiota affects his sexual choice, which is where the money's going to come from someday. But really it's becoming better appreciated that we need to understand this consortium of bacteria in our intestines to understand our own physiology. Now for most people actually, despite the sort of large numbers of bacteria, their microbiotas are dominated by two phylum, which are the Firmicutes and the Bacteroidetes here in this blue and kind of, I don't know what that color is, but I'll say pea green. But within people, there's actually tons of variety. So bacteria in my intestines today are more like the bacteria in my intestines yesterday than mine are probably like yours. So really there's a lot of individual variability in these bacteria. But despite that, actually, if you look at the functional categorization as this is done, so this is a metagenomic study looking at just basically the classes of genes in the microbiota. If you look across people, these things are actually pretty consistent. So that means essentially even though the bacterial species and sometimes genuses or phylum are different between people, really they're doing very similar things across them, meaning that there's probably a lot of selective pressures to figure out different ways to get to the same endpoint. I guess it's kind of inside out, but this is basically meaning that there's a lot of different ways to skin a cat, but it's skin inside of your intestines, so it's like endothelial. Anyway. So with that in mind, actually, if we go back, if you look at some of these classes, there's things like carbohydrate metabolism that really belie certain gene functions that are especially important for bacteria in our intestines. And actually to that, I'll say that one of the things that's been best appreciated as a mediator and moderator of our intestinal microbiota is diet. So we've known for, I guess, probably a couple of years that different bacterial phylum, sort of on the grand scale, might be differently correlated with different diets. And usually those come from studies where you're looking at two very, I guess, distinct populations. So it might be an African population, a rural African population compared to Italian children, or it might be sort of these more ancient groups of people like Amerindians in Venezuela. But we really started to appreciate recently that diet can have very, very quick impacts on microbiota. So this is actually a pretty interesting and kind of, I think, funny study, and pretty recent, where basically we've had people from various diets and backgrounds shift either a plant-based diet, which is basically a quasi-vegan diet, or an animal-based diet, which is high on cheeses, meat, and dairy products. So this is actually, this top graph looks at people switching these diets in these green bars and looks at their fiber intake. So not surprisingly, if you go on a plant-based diet, your fiber sort of shoots up. If you go on an animal-based diet, that drops to almost zero. And then what they did was they measured different metrics of bacterial diversity in those people's microbiotas. And what they saw essentially was that once the animal-based diet gets to, this is on the scale of days, once the animal-based diet gets to the actual distal gut, which is indicated by these blue arrows, you see a very, very, very quick shift in bacterial diversity. Meaning that it's not necessarily you have to have different lifestyles for the entirety of your existence to have a difference in bacteria, but this can happen in the scale of days, which is actually kind of mind-blowing. Part of the reason this happens actually is because we like to think of, you might sometimes think of bacteria as being very simple things and living in very simple environments, but realistically actually the intestinal microbiota is really, it's lots of bacteria that are involved in complex food webs. So something that goes into one bacteria isn't going to just affect that bacteria. That bacteria may then process it into another substrate, which can then either affect host, as in the case of some of these short-chain fatty acids, or it can even affect other bacteria. And what essentially means is that a single thing coming from your diet or from other parts of the gut environment may have dramatic effects that are kind of difficult to predict on all areas of the microbiota. And one of the things we've come to appreciate also is that with regard to that, most of the well-understood dietary substrates that do have an impact on this food web come from basically aspects of our diet. So I've drawn this childishly, but hopefully you can understand this is just a vegetable. But that's where a lot of, I think it's a vegetable, I haven't seen one in a long time. But actually, yeah, so one of the most direct and also sort of most impactful ways we can influence our microbiota is through fibers in our diet. The only problem with this actually is that the actual carbohydrates that come in our intestines are pretty complicated. So this might be a little bit difficult to understand here, but essentially this is just some of the different classes of sugars that exist at any given time in our intestines. So you see they come from plants, which might come from our diet. They can come from mucus that lines the intestines. It can even come from meat. There's not a ton, actually, that come from meat, we understand. Or if you're a baby or someone who has a very interesting diet, it can even come from things like breast milk. But really, there's tons of different linkages, there's tons of different monomers that make up these sugars, and how these actually can all be broken down ends up being a very difficult task for a lot of these bacteria. Now, many bacteria in your intestines are what we call generalists, which means they can actually sense, respond to, and consume lots of different types of sugars, which kind of leads to the question of how do they decide what to break down. This is essentially being a kid in a candy store. It could explicitly be a kid in a candy store. But when you have a smaller genome and you're also faced with lots of different selective pressures, how does a bacteria with a pretty simple-ish, we assume simple, sensing mechanism, figure out what's present and what it can eat? Which actually brings me to the second part of my talk. So this is actually the part that the part that Justin mentioned was both a success, failure, and then another successes and other failures. But it actually started by taking one of these bacteria that we know is really well adapted to the gut, called Bacteroides theta iota omicron, which I'll refer to lovingly as B theta because this is a mouthful. But B theta is actually one of these bacteria in the Bacteroidetes phylum that makes up a pretty sizable chunk of the healthy human gut microbiota. So like I mentioned here, depending on which study you're looking at and also the person, it can be about 3 to 5% of bacteria that live in your intestines, and that's just in a healthy human adult. And And one of the cool things about it actually is it has this phenomenally huge capability to break down different fibers and sugars from our diet and its environment. So actually kind of put this in perspective, it has about 240 encoded glycoside hydrolases in its genome, which if you do the numbers ends up being about 5% of its entire genome. And these are all just dedicated to breaking down specific linkages between specific sugars in the intestine. And interestingly also is that these aren't just sort of scattered about randomly, they're actually organized into what we call polysaccharide utilization loci, or pools. So I'm probably going to refer to these also as pools because if this is a mouthful, this is also going to be one. But essentially these are just basic gene units that are co-transcribed and co-regulated that are then involved in sensing, importing, and breaking down specific sugars. So these actually look kind of like this. So you do a very, I guess, basic schematic of one. This is the outer bacterial membrane. This is the inner membrane. And essentially what these genetic loci will look like is a set of genes that at least at the very minimum has to encode these Sus-C, Sus-D paralogs. So Sus stands for starch utilization system, and it was one of the first, I guess, one of the first poles that was found in B theta's genome, and it actually binds very specific, I guess, carbohydrates with linkages. But these Sus-C and Sus-D genes are on the outer membrane, and essentially they bind to specific carbohydrates, and they're involved in importing them into the periplasmic space. Now, this is kind of a basic schematic again, but once inside or even outside the outer membrane, glycoside hydrolases can then break these carbohydrates into smaller, more easily managed and processed subunits, and then can be further broken down so they can be imported into cytoplasm, or they can basically be shunted into metabolic processes. Now, one of the important things to mention in whole organization is that most of them will have some sort of annotated sensor or regulator, which you can imagine is really important because you don't necessarily want to have genes on all the time if you're not using them, especially in an environment like the gut where it's huge selective pressures and sort of changing environmental conditions. So these sensors are really important for sensing whatever these all process, and then upregulating the genes so that the bacteria can respond appropriately to its environment at any given time. Various specificities for different carbohydrates, but interestingly, if you grow B theta in a complicated media, complex media with lots of different sugars, we won't just upregulate them all at one time. So it doesn't sort of just take all the sensory inputs and respond immediately. What B theta actually does is that it'll upregulate its poles sequentially. So this is actually, hope you all can see this, but this is a heat map looking at transcriptional levels of B theta's poles while it grows in rich media called TYG. So if you look down here, this is a growth curve, and each of these dots, basically these poles are sampled, put on gene chips within measure transcription levels, and if the genes are upregulated, it'll look red in this heat map. If they're downregulated, they'll look green, and if they're basically not differentially regulated at that time, these will be black. So what you see initially is that once B theta goes from basically these first two dots, some poles will be turned on very brightly, while others remain pretty transcriptionally dormant. But then as you go on, actually more poles are turned on until the initial ones are actually turned off. So again, B theta is responding to its environment as it goes along, as different substrates are being depleted, as its attention shifts away from things that, or shifts to things that were once lower down in sort of pecking order. And eventually, again, more genes and more poles will be turned on. So these may be, if you want to think about this, this could be sort of a preferential hierarchy of response for B theta when it has lots of options. Now, I mentioned sort of connection between sensing and response, and this is going to be really hard to read, so I'm going to try to focus some things on you. But one of the things that was appreciated about B theta when its genome was sequenced in 2003 is that in addition to having a huge saccharolytic capability, it also has a pretty large number of different sensors in its genome. To make it a little easier to see, is that in addition to having, for some of you who are familiar with bacterial physiology, these may look less incomprehensible, but in addition to having lots of genes that are annotated as two-component system sensors or response regulators, it also had this kind of unfortunately named group of proteins that hadn't been seen before at the time called one-component systems. They're not actually one-component systems, and I won't really get into it, but it's become this huge controversy that no one cares about except for me. But perhaps more appropriately named, actually, are annotated as hybrid two-component systems. So in B theta's genome was sequenced, again, like I mentioned before. They annotated about 32 of these, and then more recent studies, including some actually in a class that I TA for, and then Casey and Justin taught, they found that there's probably 36 of these in the genome. And since being found in B. thetis genome, they've actually been found to be throughout the Bacteroides genus, throughout the Bacteroidetes phylum, and then depending on how you define them, found pretty widely throughout the bacterial domain. So to kind of explain why the unfortunate naming came about, I'm going to just distinguish between two-component systems, which have been well studied throughout the history of bacterial physiology, and then hybrid two-component systems, which are way cooler. So I'll draw these at first as dimers, but for animation purposes, I'm going to reduce them to the monomeric components. But essentially, the two-component system, which I may occasionally refer to as classical two-component system or canonical two-component system, they're made up of a two-protein phosphorelay, where a ligand binds to a periplasmic sensor kinase, leading to the internal phosphorylation of the conserved histidine residue on the sensor kinase. That phosphate is then transferred to a second protein called a response regulator, and then that impacts some sort of cellular event. So oftentimes this activated response regulator will act as a transcription factor and upregulate specific genes that are involved with dealing with this ligand. Now if you notice, there are two components in this, hence two-component systems. And hybrid two-component systems, which again are also probably dimerized all the time, have a very similar sort of mechanism of activity, except for instead of being separated into two different proteins, they are contained in one polypeptide. Interestingly, in B. thetis genome, all of the hybrid two-component systems actually also have an annotated ARC-like DNA binding domain, which I'll get back to in a little bit, but it leads to kind of an interesting idea that these proteins are initially located to the periplasm, or to the inner membrane of the bacterial cell, but then simultaneously have an area to bind directly to DNA. So theoretically, we think that they will think, and I will show, these are involved also in affecting gene regulation in response to whatever this ligand is. Now, when we first started this project, we had actually, Justin mentioned, basically one studied hybrid two-component system, one and a half well-studied hybrid two-component systems. So initially, it was BT1754, which binds to fructose, and then also early on, work from Erica actually found that BT3172 binds to N-aminosides. Now, since then, we've actually gotten some decent ligand binding studies that show that this other hybrid two-component system, BT0366, binds to a specific subset of plant polysaccharides that come from a sugar called arabinin, which is actually really common in plant cell walls. And then also we have another crystal structure of another hybrid two-component system, BT4663. I will probably not come back to most of these except for these two. So if you could remember 1754 and 0366, it'll make everyone happy. Actually, you don't have to remember, I'll tell you right now. So in addition, if you notice, actually, these hybrid two-component systems, their sensors all bind to various types of carbohydrates. And in addition to that, actually, the vast majority of them in B. thetis genome are found encoded within or next to holes. So as I mentioned before, 1754, which binds to fructose, is encoded right at the 3' end of a pretty well characterized fructan utilization pool. So these genes are all involved in processing fructose-containing polysaccharides. And another example of a hybrid two-component system being encoded for inside a pole is 0366, which is actually encoded in the middle of what seemed to be at the time an arabinin using pole, which again, as I mentioned before, seeing that it binds some subset of arabinin, it kind of makes sense that it would be responsible for upregulating the genes involved in degrading them. Now, because of this positioning and also sort of the seeming, I guess, physical restrictions of hybrid two-component systems, they seemed like they might actually be pretty simple regulatory proteins. So it seemed like at first these might be just sort of leading to, I guess, regulating these genes directly adjacent to it, but not really having global bacterial effects. Which really led us to our, I guess, real question of this project, which what are the true regulatory domains of hybrid two-component systems, and how can we sort of parse that out from overall carbohydrate processing? Now, some of the studies that, some of the classical genetic studies that are normally used in situations like this are kind of suboptimal for studying actual, for proactively studying sensory regulatory units. Part of the reason is because for a lot of these, if you want to know what genes aren't being turned on. You have to be able to turn them on and then not be able to turn them on. So if you're trying to use a knockout, you have to know the ligand for a hyper two-component system is to make sure that knockout is actually affecting those genes. For when we first started the project, as I mentioned before, basically we had ligands for 1754, the fructose binding one. We had a crystal structure for that. And then we also had some fuzzy data for 3172 and some fuzzy data for 0366. But we didn't really have direct proof that those are like the only ligands and we had the exact subunits and how those are binding. So it's a little tricky to actually get by this without necessarily knowing tons about the thing you want to study. And if you know tons about the thing you want to study, it becomes a little less interesting to study. So, I mean, that's not what science is about, right? You want to be curious and confused all the time. Another actual big problem that we came across was that if you knock out a hyper two-component system, you aren't just knocking out signaling of a hyper two-component system. You're actually knocking out all the downstream processes involved with that signaling. So in the example of fructose, if you knock out 1754, not only can B theta not sense fructose, but it can't process fructose. Bacteria it's really well understood to do stuff like metabolite repression, where basically through some other mechanisms, just straight up metabolism of different dietary substrates will have impacts on genetic expression. So it becomes a little trickier if you're using a knockout because you're kind of removing that entire area of bacterial physiology rather than directly probing and sensing. And another thing that's just a technical thing is that if you knock something out, you can't grow bacteria in that. So if you knock out 1754, B theta doesn't grow well in fructose. So just as a technical note, I'll try to make this easy because I know that for people who aren't intimately aware or care about the B theta genome, the numbering can be kind of weird. So for the rest of this actual talk, anything that's red will refer to genes in 1754 fructose responsive pull. So I just think of red, high C, or like if you want Hawaiian punch, I don't know, I think there's a regional difference. And then things that are involved things that are involved in the BT0366 associated pull will be green. Okay, so just as a sort of orientation thing. So one of the first things I did in this project actually was to look at basically how B theta would respond to different sugars transcriptionally. So this is just qPCR data looking at expression of BT0366 associated pull or the BT1754 associated pull when growing B theta in different sugars. So if you look here, this is wild type B theta. You grow in fructose, you turn on the red gene, so the 1754 associated pull. If you go into ribonane, you turn on the 0366 associated pull. And importantly, actually, growing in fructose doesn't affect 0366 appreciably, and then growing into ribonane doesn't affect 1754. So it looks like, again, there's some sort of sensing specificity for response in these different sugars. Now, if you knock out 1754, as I mentioned before, you actually can't grow in fructose, so these studies become not meaningful. But it doesn't affect the ribonane signaling again, so you still get a couple thousand-fold induction of these pulls. And then finally, if you knock out 0366, you can get some decent growth in ribonane. I guess it's kind of hard to see here because it's not the purest prep of a ribonane, so it's probably B theta is growing in other sugars. It isn't upregulating. Importantly, it does not upregulate the 0366 pull, but again, it does not affect expression of the 1754 associated pull. So with that information, actually, what we can do is we know that we have a pretty well-characterized sensor that binds directly to monomeric fructose in 1754. And we have, at the time, actually a pretty poorly understood regulatory network of 0366. So what we can do is take that sensor, like Justin mentioned, glue on a disguise, although the mustache was a little gaudy, so I don't know if I'd go with that. But take that sensor, glue it onto the response regulator of 0366, or the regulatory domains of 0366. Then we can grow this construct in fructose, and then see basically what genes are turned on when we activate this chimeric protein. And really the questions we want to ask are kind of, can we verify that 0366 is responsible for upregulating the adjacent pull directly? And then kind of more importantly is that, can we turn on genes that aren't necessarily directly in the physically associated pull of 0366? So just schematically, what it looks like in our first construct is we have the endogenous 1754 here, and then we have a distally inserted chimeric hyper two-component system at another. Importantly, it's regulated by the same promoter, but is actually a distant point in the genome. So as Justin mentioned, if you take this construct, so this B theta construct, and you grow in fructose, you get normal upregulation of the 1754 associated pull, but it really doesn't do anything for 0366. At first glance, what this means actually is that our chimera didn't work, which is fine. It was fine like when I first started, but had this happen now, I would have been crying. But actually, importantly, led us down a pretty interesting avenue, which is that this is just wild-type B theta, and it's possible that fructose signaling or fructose sensing could actually have a negative effect on O366. So basically, fructose would be not only upregulating its normal genes, but also repressing the genes our chimera would be inducing, which is ended up being what we found. So this is just qPCR again, looking at gene expression of B theta, wild-type B theta, when it's grown in fructose. Again, those genes aren't induced. Arabinine, turn around a couple thousand-fold, or both. It's interesting, if you grow in fructose and arabinine, you get about a 60-fold repression of the O366 associated pull, meaning again that fructose signaling is having some negative regulatory effect on these genes. However, luckily, is that we can relieve this repression by knocking out 1754. So basically, by ablating the ability of B theta to sense and metabolize fructose, we can then grow B theta in both of these signals and have the genes turned on. Interestingly, actually, this repression is also, I guess, it shows through when you're looking at just at carbohydrate consumption. So this is HPAC data, looking basically at the sugar content of media of B theta grown in either fructose or arabinose, or both. So this is actually, arabinose is the subunits of arabinine, but is basically used as a proxy for arabinine here. So if you take wild-type B theta and you basically survey the media over time, you see at first there's both equal amounts of fructose and arabinose in this media, but then actually pretty quickly you'll deplete your fructose while leaving all the remaining arabinose there, essentially showing that B theta preferentially consumes fructose in addition to responding to it. However, again, if you knock out 1754, so B theta can't sense fructose, now you switch B theta's attention to consuming arabinose while leaving the fructose remaining in the media. So it's just what we end up doing. Instead of leaving the endogenous copy of 1754, we knocked it out and then added our chimera. And now we can grow this construct or this strain of B theta in fructose and actually supplementary sugar, so arabinose in this condition. And we find that if we add fructose, now we can basically mimic signaling of the endogenous O366, which is basically like, I realized when I was going through this yesterday is that I just spent about 8 slides showing you what we already knew was going to happen, which is if you turn on the regulatory domain of O366, you can upregulate the O366 associated pull. And then I sort of had a breaking point, and I just started weeping and weeping. And this morning I was still weeping, and I was super dehydrated. But I came in anyway. So I mentioned before that we want to get an idea of the global impact of these proteins. So maybe the problem was that we were looking at too close an area. So if you look at the O366 associated pull, okay, we can turn that on, but does anything else happen elsewhere in the genome? So this is a little bit confusing just because the colors are slipped a little bit. But again, the O366 pull is denoted by this green bar and these genes up here. The 1754 associated pull is denoted by this red bar and these black genes here. This is just a heat map looking at any significant genes in B theta's genome that are transcriptionally regulated when we have our activated chimera there. So I'll walk you through a little bit more of that. But essentially what happens is that if we activate our chimera and our conditions that aren't repressing our chimera, we get a really strong upregulation of the entire O366 associated pull, again confirming that basically O366 is responsible for upregulation of its adjacent pull. And importantly, actually, we don't get any regulation of 1754. So our chimera is acting explicitly as the regulatory domains of our protein. Basically, we've shifted that completely to being a mimic for O366. Now, this in and of itself, as I mentioned before, wouldn't be the most exciting thing. But really importantly is we noticed that there's also several pulls that are located distally on the genome that are actually downregulated when we activate our chimera. So these 3 pulls, which luckily having a genome with lots of numbers, you can tell these are actually far apart. So this is O366. These are about 2,000 genes away, similar here. But when we activate the O366 regulatory domain with our chimera, we also end up repressing 3 pulls that are involved in mucin degradation. So I mentioned that B theta has a wide saccharolytic capability, and in addition to breaking down different parts of plants, it's also really adept at breaking down different parts of mucus. So essentially what this means is that in these conditions, if given a choice, B theta will respond preferentially to plant material while not responding to mucus. Now this is really cool, and we kind of wanted to see if this would be a more direct interaction. So what we did was a chromatin immunoprecipitation followed by high-throughput sequencing, or ChIP-seq. And we used our chimera, cross-linked our proteins to our DNA, then used antibodies against the periplasmic domain of our chimera, and basically saw what was being pulled down. So we kind of wanted to see if these hybrid two-component systems are actually directly binding to any DNA, and if so, where that was. Now, upon this, what we actually ended up finding was that O366's regulatory domains not only bind to the genes upregulated, so it's associated pull, but it also directly binds to the genes that's responsible for downregulating. So what that's it's kind of a curious finding, because essentially it means that it's binding directly to its positive and negative regulatory targets. Now, if you ask me a bunch of questions about this, I had a bunch of really crackpot theories that ended up not panning out, so I won't go through them, but we think that there may be other accessory proteins or other conditions that may be allowing B theta to act in different means in response to different pulls. So it ends up as we have a model where basically B theta is in the intestinal environment and gets lots of different signals. So these would be, for example, a hybrid two-component system binding to a sugar, and then some other sensor binding to a different sugar. Both of these will have a positive regulatory impact on their respective pulls. But at the same time, the hybrid two-component system will directly repress other pulls, and it'll do so in all these cases by binding directly to the genome. So as I mentioned before, it ends up being basically the creation of a carbohydrate consumption hierarchy, where B theta has preferential responses to certain sugars when given options. So in our case, what we found was that these plant polysaccharides lead to repression of mucin degradation. So I think that's pretty interesting. Basically, B theta was given a supermarket, and it went to the health food aisle rather than, I don't know what mucus would be. Hopefully you can't buy tons of it at the store. So I'll now talk a little bit about some stuff that I think we're really excited about, has been difficult and really useful intellectually in terms of my growth. So I'm going to show it anyway. Again, there was no tears left, so it's going to be. But if you notice, there's we have kind of a spatially difficult situation to work through right now. Because what we have is a periplasmically, or I say, intermembrane localized protein. It spans into the periplasm, but also directly binds to DNA. So we actually showed that the hybrid two-component systems aren't actually broken down. That's one of the things we thought at first, was that you could have something expanding the periplasm, but then once it gets activated, it breaks basically into a two-component system. But we found that actually by pulling down the periplasm in our ChIP-seq experiments, we could still get DNA. So that means it seems like that unit remains intact, at least physically associated with one another. But, sorry, I said that. But actually, given that B theta has dozens of hybrid two-component systems and can respond to dozens of different signals, and also theoretically has some sort of other genomic organization, this becomes kind of it could become kind of a tangled web when you'll look at it. And even if it's not a constant tangled web, it could be a dynamically tangled web that might be, it's a very important question for bacteria to solve. So basically, how do I organize my genome in response, and how do I organize my genome with respect to my responsive capabilities? That's something we want to understand better if we want to understand how B theta is sensing and consuming different sugars. So the first thing we did is actually just develop some microscopy techniques for visualizing B theta. I didn't mention this before, but B theta is actually, it's an aerotolerant anaerobe, so it won't immediately die when grown in atmospheric oxygen, or when placed in atmospheric oxygen, but it won't grow. So what we had to do actually first was figure out a way to contain B theta in little anaerobic environments so that we can then put it at 37 degrees and watch it grow, which is more, it could have led to some really cool stuff if we could do some anaerobic imaging, but this is just B theta being grown anaerobically under a microscope and dividing over about 4 hours, I think it was. Now the next thing, once that happened, we said, oh, we figured out ways to do this. Now if we can image these things, we can tag and localize different proteins and parts of the genome. All of a sudden we have more insight into how these things are being organized. So one of the things we tried to do was to pop a GFP into the 3' end of the endogenous O366. Then what we did was we grow B theta in a rhabdonin, so we try to induce O366 signaling, see whatever it's doing, and then image it under a fluorescent microscope. So the part about being an anaerobe is important because GFP, which you notice isn't the ideal tag, requires oxygen to actually mature. But unfortunately, at the time, it was actually the only fluorophore that we had working in B theta. So when we started this project, we've gotten a pretty awesome construct, or a pretty awesome strain from Eric Martens at the University of Michigan, which constitutively expressed GFP and therefore could be seen illuminate, or could be seen fluorescing when put in oxygen. We tried some anaerobic imaging techniques that just didn't pan out right now, but I can talk about this more later if you'd like. So these are going to be really hard to see. I'm doing it. So these boxes actually aren't anything particular. I just wanted to sort of highlight groups of cells. But essentially, yeah, what you're looking for here is puncta. It's a little fluorescent puncta representing GFP tagged 0366. These were movies. They're really hard to see. Also, I should mention that this is all done with help from people from Casey Wong's lab and Julia Terrier's lab. So we have Tim, Carolina, Tristan, and billions others in Casey's lab. And then also Rick Zuzo, who helped me basically determine the autofluorescent spectra of B theta, which unfortunately, making GFP even worse, is right in that green cyan range. All lessons learned. This is just another construct doing the same thing. But as you can see here, if you look really carefully, there are certain puncta that are developing and stagnating. Sometimes you'll see them sort of flicker in and out of frame, and we don't know if that's turnover or if that's sort of movement of proteins, but something we'd like to follow up on. Now, our strategy for labeling 1754, our other sort of hybrid two component system of interest, is a little bit different. So instead of popping GFP into the endogenous 1754, what we did was we complemented a deletion of 1754 with a tagged version. So deleted 1754, and then at that same distance site that I mentioned earlier, we put in a GFP tag 1754. So these arrows are sort of just highlighting, but most of the cells in here are actually showing puncta. So you can see kind of a little dancing. And these are, again, grown in fructose, so they should be stimulating 1754 activation, at least as far as we understand it. Again, here, the arrows, again, pointing to little puncta. You see sort of running up and down these cells. Now, I mentioned that this was sort of a difficult, I was flying high trying to do this microscopy stuff, and it ended up being a little more difficult. So we've had the problems of having to use an oxygen-dependent fluorophore and an oxygen-intolerant species. We've also had some difficulties. Both of these constructs are hypomorph, so they don't fully recapitulate endogenous protein activity, which we've been trying to alleviate by basically putting a tether between our protein and our tag. And I should mention also that for a variety of reasons, we've tagged 1754 and 0366 at the 3' end, which is where the DNA binding domain is, which could have a huge effect on how they're actually localizing the cell. So we're hoping by adding that tag and possibly by moving the fluorophore to a different part of the protein, we may also reduce some of the impact we're having on normal protein activity. So the next thing I'd like to do is actually look more at the genome, in addition to looking at how hybrid two component systems are moving. So I mentioned we'll try to improve basically our tagging techniques for the hybrid two component systems themselves. And then also, in addition to seeing where they're individually located, seeing if there are sort of organizations within the membrane. So it's possible that each of these is independent systems. It's also possible there's sort of like a hybrid two component system raft in parts of the genome. So it might help spatially localize the genome if the hybrid two component systems are also spatially localized to specific areas rather than having to be free floating. We're also working on trying to directly tag the genome using fluorescently tagged LAC-I and TAT-R proteins. And it's another thing that actually might be very interesting, likely will be very interesting to see, even independent of hybrid two component system activity, whether or not there is some sort of spatial organization of the genome and different conditions. So for a species that dedicates 5% of its genome specifically to glycoside hydrolases and about 18% to pulls, really, the positioning of the genes has got to be very important for how the bacteria overall is functioning in the gut environment. So just really briefly, I'm kind of like to go through like just a conclusion. So conclusion and slight prospectus. So we ended up finding in this project was 3 distinct tiers of B theta's preference for carbohydrates. So at the top here we have this monomeric fructose, which for a lot of American kids also is probably at the top. Yeah. But essentially, this is something I didn't show data on this, but not only does 1754 signaling of fructose binding important for active or for putting at the top of that hierarchy, but also, as I mentioned, catabolite repression is really important in this. So we can actually alleviate some of fructose's repressive capabilities by either knocking out 1754 or by knocking out a fructokinase, which is one of the first steps in putting it into central metabolism. So it looks like fructose is actually imparting its repressive capabilities in several different ways that are all sort of partially redundant, but also maybe affecting different parts of physiology. The next up is this really sharp plant material. So in our case, it's arabidin. So again, we have monomeric sugars at the top, fructose, and the next would be actually plant materials, which probably come from our diet, hopefully. But it's decidedly below the fructose response in B theta's physiology. And then finally, we have host mucus. So in this case, we actually used a single component of host mucus. But again, the response to host mucus by B theta is definitely below arabidin and fructose in terms of its sort of preferential response. And finally, I just try to go through why we think this is important to study. I mean, it's interesting to think that bacteria make decisions or have preferential responses, but sort of why is it important to look at? So one reason is that we found that hybrid two-component systems are not just a sort of freak accident that happened in B theta's genome, but it's really spread throughout many bacteria in the intestines. And it looks like they're having a pretty large impact on how these bacteria are acting. And since we know from the beginning of my talk, that basically the microbiota is this huge sort of wild west of impact for our own physiology, really understanding how this really widespread protein is quite important for understanding how we can manipulate the microbiota. So another thing that's actually kind of might be interesting to look at is that we studied one hierarchy in one species, but we don't necessarily know if that hierarchy is universal or if there's actually selective pressures that cause bacteria to have different responses and different sort of preferences for sugars in the gut. So understanding how bacteria preferentially choose different sugars could give us an insight into how these things are living in the intestine. If we find a species that preferentially responds to mucus, it could mean that that bacteria is more, is more intimately involved in consuming mucus than B theta. Versus a bacteria that only responds to plant materials, it may indicate that that bacteria is more, distantly related to involving mucus. And finally, actually, this is something we try to put in most of our grants. But essentially, if we want to manipulate the microbiota through our diet, which we know is possible, and it's actually pretty powerful as a tool for manipulating the microbiota, we have to understand how we can actually manipulate that diet to have the biggest impact. So there's this field of prebiotics, which essentially is the administration of specific dietary substrates to preferentially expand certain bacteria while not expanding harmful bacteria. And that's become something that's really well appreciated and trying to become better studied, but it's still kind of mysterious for us. But really, what our findings show is that you can't just add one substrate and expect it to have one big impact on specific species, especially if the rest of the diet isn't controlled or the rest of the environment in the gut isn't controlled. So in order to have an intentional impact, we have to really think about what the entire surrounding area for the bacteria would be, and in order to get an actual response, or in order to predict how it actually respond. And with that, I'd like to just give a few sort of professional acknowledgments. So of course I would thank everything in the Sonnenberg Lab. As Justin mentioned, I've been here as long as many of them, and so they're like my babies. Like the babies I can't give away. But these are actually, so when I started the lab, these are a few of the people that sort of helped me get on the ground, including Wes, who's one of the newer additions, but has been helping me a lot with the cloning aspects of the imaging project. As I mentioned before, a lot of people in Casey's lab who've been helping me try to get microscopy off the ground, and then also in Manuel's lab, who have helped me try to do stuff like immunofluorescence. People in Julie's lab, like Rick, who have helped me identify specific autofluorescence spectra of B theta, which have been hopefully help us more, I guess, more intelligently choose fluorophores and imaging techniques in the future. I'd like to thank Josh for agreeing to be in my chair today. I'd also like to thank Eric Martens and David Bolam, who have been really, really useful experts on both bacterial beef theta's nutrient capabilities and also in hybrid two-component systems themselves. So they've been really useful sounding board, especially earlier on in the project. And I'd like to thank people at UCSD and Duke for helping us with the HPAEC and ChIP-seq experiments. I'd like to thank the administrators in the M&I department who have been super useful in just making sure that I don't, let's say wander off into the ether, but it wouldn't be a wandering, it'd be sort of just sitting down to ether surrounded me. But it really kept me and the rest of the department running like a pretty well-oiled machine. And then finally, I'd like to just thank my funding for helping us do a lot of really cool experiments in the past couple of years. And with that, I'll take any questions you have. Yeah.
Of course, we did it with rapid fructose to affect fitness function of some wild type.
So experiments like that have been used more in mice, and then it does seem like you knock out capabilities. So if you knock out 3172, which is another hybrid two-component system, you definitely do see a fitness defect. With 1754 and 0366, so fructose and arabinine, it looks like if you grow B theta in either one of those, it grows up, at least in culture, very well. It's a little different, actually. The mucus carbohydrates B theta can grow in, but not very well. So there it looks like it's a little more clear fitness benefit to use one or the other. And that's kind of one of the things is we thought, the hierarchy, is that hierarchy, well, not is that hierarchy, but odds are that hierarchy is not just sort of put together by chance. So it looks like, at least in the assumption, there would be some sort of benefit for using fructose over arabinine. But then the benefits of that could be, it could be a little fuzzy. It could be that energetically it's just easier to sort of pop into metabolism, that's great. That could be that you have to do less processing, so you have to produce fewer enzymes to be able to degrade it. It could also be that, so I guess fructose is probably more transient in the gut because it's more easily consumed, and also it's probably not as abundant because it's being absorbed higher up in the gastrointestinal tract. Whereas arabinine, if it's coming through plant materials, it would be transiently present, but might be more, I guess, commonly present if you're eating plants. Whereas mucus is kind of always there. So there's a bunch of not appropriate metaphors that can be used, but essentially that's just sort of like with anything, if no other options are available, then mucus will be there, and I don't grow that well in it, but might as well give it a shot, sort of thing. Please.
You mentioned that the poles are activated sequentially.
Mm-hmm.
Is that just due to priorities and abundance, or is the signaling network somehow set up that it has time to go through sequential time?
I don't think it's dependent on time. Like if you sort of more intentionally organize that, if you more intentionally organize the complex environment, you won't necessarily turn on all the poles in the same order. You'll get skipping of different rungs. And I'm trying to think of any other examples I've seen where time is explicitly ruled out. But that's my feeling, is that it's not just sort of like a clock. It's not like you're hitting a timer like once for
Time is
yeah, and I think it's also kind of intricately tied to presence and depletion of things. So actually, I guess I didn't mention this, but in the HPAEC experiments, where you're measuring just monosaccharides left in the media, you start seeing arabinose being depleted towards the end of that, but it seems like it's dependent more on the depletion of fructose first, and sort of just, again, clicking off when that sensing isn't as strong. Yeah.
You mentioned something, maybe I didn't get it right, but you mentioned something about product-based repression.
Mm-hmm.
Like if you're using fructose as a product of that represses the operon.
Mm-hmm.
Why would you do that? I mean, if there's a lot of fructose around and you like it, why would you shut down the pathway?
So
is it repression of another operon?
Yeah, so it's typically a catabolite repression. It is something that's found in bacteria, but it will either sort of dampen its own response, so it could just be like a negative feedback loop, which seems like it's more, I don't know if it's this is how I understand it, but it seems like that's not so much like turning off, it's not turning off things as you see them, it's more of just like detecting abundance of something and not necessarily putting too much work into consuming more of it. So in a lot of bacteria, it's more tied to cyclic AMP levels. So it could be, if we have enough cyclic AMP, let's not put any more work into making more. And then with
you don't have to upregulate the enzymes anymore because there's plenty of substrate. Exactly,
yeah. And so that's kind of what we thought initially with B theta, was that it was doing something similar to that, except for it doesn't have cyclic AMP. But as I mentioned, even if B theta can sense through 1754, there's still some repression that is mediated by fructose metabolism. So it could be something like that, but we don't know exactly the mechanism of that specific area of repression.
I was wondering if it were food, maybe I should have a little bit of
just a touch, yeah. Yeah, that'd be really sad actually if intestinal bacteria were like somehow more responsible adults than like humans. But it's definitely a possibility. I mean, you have that much selective pressure, it's possible that they. Yeah.
Are there ingredients associated with aberrant levels in beer? It's too high or too low?
So theta or other things, but it's sort of associated with, a lot of times you'll see sort of correlations with, some taxa or genus, some phylum or genus, and certain diseases like IBD or obesity. And so things, I guess some of the better examples are comparison of children from Burkina Faso with children from Italy, found that basically people in this Western diet tend to have higher firmicutes and also higher Bacteroides. So higher Bacteroides relative to Prevotella and other Bacteroides species. So there it's kind of hard to tell chicken and egg. It could be that if they're eating more fat and meat and less fiber, that these are just somehow expanded. But in those situations, you also have problems running into genetics and different steps of lifestyle. So B theta itself, I don't think has been directly associated with any specific disease, but sort of skewing towards B theta or other Bacteroides maybe. Other related species, so B fragilis so Bacteroides fragilis is another related species that is sometimes associated with some diseases, especially if you Google it. So sometimes it's been found in abscesses in the intestines, but then also on the other side, it also has a really profound anti-inflammatory effect. So it's probably not very clear-cut to which, none of these are directly causing sort of postulates of causing disease, but they may be associated with it.
Based on your ChIP-seq data, were you able to determine the DNA binding sites of these glucose systems? And does the location of the binding sites affect whether it's an activator or oppressor of certain operands?
Right. So we weren't able to directly find it from ChIP-seq. Like basically, we looked at ChIP-seq and tried to find any sort of consistent sequence around there that might explain why that heterogeneous component system was binding to each of those different spots, and then looked for stuff like sort of like you said, like if you could find some sort of positive signal or positive co-sequence and then positive or negative co-sequence. I didn't end up finding any sort of sequence similarity, any obvious sequence similarity between any of those sites. So there's some spans where you might get like 10 nucleotides in a row that were similar, but it's not something I was confident in saying. One thing that's actually kind of interesting, and I didn't mention it, was that all those binding sites are actually downstream of the poles they're regulating, which that was one of the rabbit holes I went down and started figuring out how that could maybe explain. We thought there might be some sort of genomic rearrangement or flipping or things like that, or other sequences being inserted. But long story short, we didn't find any signatures. And the only thing that's kind of close is that we found there is a tandem sequence at the 0366 associated pole binding site that looks like it could actually be sort of enough space for 2 dimers to bind. That's something I'd like to experiment, or I'd like to experimentally verify before calling it a binding site, although there is a paper that has identified that binding site by just looking at sort of anything that's repeated. So for those of you who are interested in sticking around and seeing other parts of my life, I do have, I mean, I wouldn't, sorry. But I do have some personal acknowledgements, so it could be fun. So find them first. So I think at different parts of my graduate career, I've required different levels of support and love, and usually it's high to very high. So I'd like to begin by thanking again each and every, pretty much everyone who's ever touched my life. I think this. I'll explain that more in a second. But I'd like to. So the only thing is that even though I love all of you very much, I don't take tons of pictures. So I had to resort to some pretty desperate measures to get anything. So speaking of Nikki, I'll start off with Justin. So, this is an artist depiction. But so Justin mentioned I was one of the first people to join his lab, and it's been an amazing experience to have you as a scientific mentor, as a friend, as a person standing next to me on screen. But I can't imagine, I can't imagine having so much support, especially early on in my career, and how much it shaped me. So I can't thank you enough. And actually, this is how much I'd like to thank you. This is water, I'm sure. Yeah, I was actually kind of upset you took like half my pictures. So, I got most of these off my sister's Facebook, so I'm not sure how that really makes any sense. So this is kind of what the lab looked like when I started. Actually, shot for shot. It was really small, and actually I joined a day after. But it's really cool because I've joined so early on, we've gotten to see so many people join, and now people start to leave. It's been really fun to see the lab transform, both scientifically and sort of personally. So that's been a lot of fun. It's been a fun train to ride. Yes, this is actually kind of what it looked like. And actually, so I started so long ago that this is what we looked at. This is what we looked like, sort of. This is actually probably not even around super projects, so you can probably date this well. But when I started, Justin and Erica actually looked like this. I don't know if Erica knew this, but Justin actually used to date Selena Gomez. But actually, this is the lab. This is more what the lab looks like today. So I won't necessarily call it everyone in the lab because I was told to. I don't play by the rules. But you can try it if you'd like to. I can point you out later on because I think it'd be kind of funny. But it's really cool. You can see the lab expand and seeing tons of different personalities and different hobbies, stuff like Beer Club, and even stuff like the retreat, which I don't have any pictures from that for some reason. Like, I think legal reasons, but it's been tons of fun to see and get to talk to people too. Even lab meetings, apparently I'm the confrontational one. But even that's been a fun process for me, just getting to talk science to really smart, really fun people. So thank you for all being there. If only to point out my spelling mistakes that I probably made thousands of. As I mentioned before, this is one of our sexier moments. Not intentionally, but just by the Power Rangers. Like, come on, it's Power Rangers. And this has been tons of fun stuff we've done, and something that I think I will have trouble duplicating elsewhere, hopefully. These are just a few of the sort of organizations and people that have been really supportive for me throughout grad school. Grad school is tough. It's supposed to be. If it wasn't, it wouldn't be grad school. It'd be a higher paying job. But this has been something that's been a lot of fun, and I really like just the fun and excitement and just sort of enthusiasm for life and supportive people that I can't thank you enough. Also, as Justin mentioned, thank you to the hospital for Humpty Dumptying me back together. Because there were times when holding tubes is a little difficult, but made it. This is a non-comprehensive list of or pictures of people that have been just sort of there to help me decompress their life. So there's people from in and around lab, there's people from in and around department, there's people from in and around being really dirty, there's people from college and high school and college. So I really, again, it's been nice to be able to get out of lab sometimes, even though Justin's in Massachusetts whenever you need something from it. But it's been, I think it's the people in your life that make life fun, so thank you for that. This is was a good start for my graduate school career to come in with a really awesome class. Guess who this one is? But this is something actually. It's really fun to see people that are like your own age become successful and like do fun stuff and also be like surprisingly intelligent. I'd call each of them surprisingly intelligent because I was very surprised by it. Like, you can tell given the way they look. That's slightly more. And in particular, I'd call my lab mate, classmate, day mate, Cat. So as Justin mentioned, we're not necessarily the best influences on each other. But I think this low level of antagonism is really healthy for both of our growth. Actually, it's been healthier for my growth, specifically in this direction, and less healthy for Cat's growth, specifically in this direction. And as you mentioned, even this sort of this became like a really surprisingly dark, like therapeutic thing for us. We're like, oh, look at me, I'm Cat, I'm falling asleep. Oh, look at me, my leg's broken, I'm John. So I don't know if that's necessarily like the most healthy friendship, but it's been a very productive and fruitful one, and I appreciate it. I'd also like thank various areas of my family. This is not I try to draw them standing on a hill. But around this one, it became a little tough. So this is family, I guess, depicting family from California, who've been really nice, and especially coming out for events and housing me for Thanksgiving and getting me drunk on Thanksgiving, and then getting me drunk the day after Thanksgiving, and then getting people to get me drunk the day after Thanksgiving, and then driving me to the airport. I really appreciate their support and sort of their warm embrace. And then I have tons of family. Actually, this is part of my dad's family in Virginia. So it's been really awesome. Again, having family that's a little further away, but also still very close to your heart. And then these are some people that have left us. Let's see here. This is my cousin Audrey, her husband Kevin, their baby, well, one of their babies now, Morgan. And this is something that actually came out for graduation, including my cousins who've sent me. This is Kayla and Keenan. They've sent me like oddly threatening like cards. So I love them, but like I'm worried because there's been a lot of like diabolical laughter written into greeting cards. I don't know how kids do that. But it was really awesome to have all my family, or a lot of my family out for graduation recently to see me wear a funny hat, I guess. Somebody say, oh. Come on, people, let's keep it together. So Christy, I'll save most of the nice things I'm going to save for a very specific day sometime in the future. But I'll just say this is our best picture. It's like, I don't know what either one of us is doing, but you can't see this. It's my little sister's street, and this is at her graduation. And you see it's like a super awesome background. Like, it was really pretty and idyllic. And then I don't know why we both look confused. Or I don't think she's even talking to you. I think she's just doing this. I'd also would like to thank Christy for the newest member of our family. Just kidding. Okay, no, totally just kidding. So, had to roll the dice on that one. Had to roll the dice. So that's not going to be smart long term, but that was kind of funny. But seriously, I'm really happy that I met you. Even this is the second best picture we have. And then finally, I'd like to thank my parents and sister. This was actually a really adorable. I think this might have been my idea, but we did a Christmas photo this series a couple years ago. So we actually came up with some pretty adorable ones, including like these. If we had lasers in the background, that would have been primo, but they don't do that anymore because they probably kill a bunch of kids. But I know without my parents and family, I wouldn't be here today. And I don't mean that biologically, because I think that this collection of genes would have figured itself out. For better or for worse, like this is either like a super disease or some sort of apex predator. But I can very easily say that without your support and guidance and love, I don't know where I'd be. Actually, without that support and guidance and love, I wouldn't have created low-level elder abuse in the past, which, again, is just kind of fun stuff we do. And even this kind of stuff, so going home to snow has been almost indrivable. So yeah, this is where I get a lot of my fashion sense. Again, I can't thank you guys enough. Besides this is probably as good as I'm going to. So yeah, thank you all for being here, and
Please come to church house in about an hour for a celebration.
Congratulations,